Hi everybody! I hope that you guys are all doing well, and I hope that this video helps you answer number 3 on your student response sheet for 3.1.3, When Cells Lose Control. So number 3 asks us to draw a feedback loop depicting the role of oncogenes and tumor suppressor genes in cancer. Before we start to answer this question, what I would like to do is go back to your introduction where you guys had to draw out the cell cycle. So the cell cycle can be thought of as like the life cycle of a cell. We start over here with one cell, and then we go through just a series of steps where the cell is growing, it's replicating its DNA, it's preparing to divide, then it divides up the DNA, and then ultimately it divides the cell completely into 2 new daughter cells. All of our cells go through this process in order to help us grow and to help us repair. So, along this way, we are going to hit a series of steps that we call checkpoints, where the cell kind of pauses and decides, did it do everything it was supposed to do within that given phase? So, for example, in G1, it needs to make sure that the cell has increased in size that it will allow it to divide, it has enough nutrients available, there's growth factors present so that the cell has what it needs in order to carry out the process of cell division, and that the DNA isn't damaged. If the cell decides that everything is okay, then it proceeds into the next part of the cell cycle. But there are some times when it gets to this checkpoint and it realizes that not everything was done. In those cases, it can do one of 2 things. It can go through what we call GO, which is basically a stage where the cell is arrested and it just stays there waiting for the cell to complete everything in G1 and then give the signal to go ahead. Or in some rare cases, there's cells in your body, like your nerve cells, the heart muscle cells, that stay permanently in GO for the rest of your life. Another option when we get to this checkpoint, if not everything is done or there's problems, the cell will get a signal to apoptose, which basically means the cell burst open and the cell dies. So these checkpoints are really important for the completion of the cell division process. So let's go back here to number 3 now. So we're going to start off with what I call normal cell division. And that's what we want to have. So we want the cell cycle to happen normally without any problems. Now this is regulated or controlled by our proto-oncogenes. You're going to hear a lot of videos or see it referred to as like the gas pedal in your car. So when we approach that checkpoint, if everything's fine, we give it the gas or the go-ahead to continue dividing. So this process happens all the time where the cell is going to stop at that checkpoint. If it's good, the proto-oncogenes are activated. They give the cell the proteins they need to proceed into the next step. But sometimes those proto-oncogenes can become mutated into what we call oncogenes. I'm going to put a little sad face because this is the bad one. We don't want those oncogenes. So if the proto-oncogenes become oncogenes, it's analogous to that gas pedal being stuck down. So the cell is constantly receiving a signal that tells it to divide and keep dividing. Obviously, if this happens, those oncogenes are going to trigger uncontrollable cell growth. So I'm just going to put leads to. Now, obviously, our body is well designed. We have a backup here. We want to stop that uncontrollable cell growth. And that's where our other set of genes comes in, called tumor suppressor genes. So our uncontrollable cell growth is stopped by our tumor suppressor genes. Keeping with that car analogy, these are like the brakes. One example of a tumor suppressor gene is P53. So P53 does a lot of different things in order to control this cell division process. One thing that it can do is it can activate DNA repair proteins. And those DNA repair proteins would be able to go back and kind of fix these proto-oncogenes to make sure that they are not sending out those signals to continuously divide. In some cases, if the DNA is just way past repair, then P53 will also trigger apoptosis. Which again is that cell death where it basically punches holes in the cell membrane and then the contents of the cell leak out and the cell dies. So tumor suppressor genes are going to stop this uncontrollable cell growth. Unfortunately, we can end up having mutations in these tumor suppressor genes too. So if our genes fail or we have mutations, it causes the tumor suppressor genes to not work. And then if those tumor suppressor genes are not working, we are not going to be able to control that cell growth. And ultimately, if that continues, then it leads to cancer. I hope this was helpful, guys. And please, if you have any questions about the material, I created a discussion board in this week's assignment. Post those questions there. I'm sure if you guys are thinking it, your peers are also thinking it. This will be a nice way for me to address those questions and then hopefully answer questions that other people are having. Thanks, guys. Have a good one.